Hey everyone, welcome to the video for Module 6.1.2. In this module, we're going to go into a brief intro into cellular electricity. You can see the learning outcomes described here. I want to start off by talking about the rest of membrane potential, which is simply the unequal distribution of charges. In this, the cytosol is highly negative compared to the ECF, or the extracellular fluid, and there's a high sodium concentration in the ECF and a low sodium concentration in the cytosol. Keep in mind that the resting membrane potential is roughly about minus 70 millivolts. We're going to talk more about the neuromuscular junction as we move forward, but I just want to introduce it here because it has some importance. The neuromuscular junction and nerve stimulation specifically. So skeletal muscle cells are stimulated by a motor neuron, and the axon of each motor neuron branches extensively to form numerous cellular extensions called the synaptic terminals. The synaptic terminals then interact with the sarcolemma of the muscle fiber at each specialized site called the neuromuscular junction. And when the electrical impulse or the action potential reaches the synaptic terminals, the calcium channels with the synaptic terminal begin to open, which causes calcium to rush into the axon terminal. And as a result of the influx of calcium, synaptic vesicles that contain the neurotransmitter acetylcholine, or ACH for short, fuse with the axon membrane, which then releases the neurotransmitter into the synaptic cleft by exocytosis. Acetylcholine then diffuses across the synaptic cleft and attaches to receptors called ion channels on highly folded regions of the sarcolemma at the motor end plate. So the binding of acetylcholine to the channels causes the channel to open, and then the influx of sodium ions into the muscle cell results in the depolarization of the sarcolemma and eventually leading to an action potential and then subsequent contraction of the muscle. As the action potential spreads away from the motor end plate and across the sarcolemma, acetylcholine is swiftly broken down by the enzyme acetylcholinesterase. The destruction of acetylcholine prevents the continued muscle contraction in the absence of nervous stimulation. And we'll talk a little bit later on in this unit what would happen if acetylcholinesterase was inhibited. So here we want to talk about the generation of the action potential of the muscle. And again, we'll get into this more detail, more in depth in the next unit. But in order to generate the action potential of the muscle, we have to first start off with electrical conditions of a resting sarcolemma, which is also called the resting membrane potential. That's considered to be polarized. Basically, what that means that the extracellular environment is more positive with respect to the inside of the membrane. The predominant extracellular ion is sodium, while the predominant intercellular ion is potassium. The sarcolemma is relatively impermeable to both ions at rest, but more so to the sodium. So in order to get the muscle to contract, the membrane potential must depolarize. So step one in that is the depolarization. So the binding of acetylcholine to the sodium ion channels on the motor end plate causes the channels to open. Sodium then comes in rapidly across the sarcolemma into the cytoplasm. And as sodium begins to accumulate on the inside of the muscle cell, the resting potential is decreased and local depolarization occurs. Basically, that means that that patch of sarcolemma immediately around the sodium channel becomes more positive inside with respect to the outside of the cell. In step two, you start to have propagation of the action potential. And that is the positive charge inside the initial patch of the sarcolemma changes the permeability of the adjacent patch, and that opens sodium channels there. Consequently, the sodium begins to rush into the membrane there and causes the membrane potential in that region to decrease, and depolarization occurs in that area. Thus, the action potential begins to travel away from the motor end plate and across the entire sarcolemma and then down to the T tubules. So we have excitation contraction coupling. As the action potential propagates along the sarcolemma and moves down the T tubules, transmission of the action potential past the triads causes the terminal cisternae of the SR to release calcium into the sarcoplasm. There it now becomes available to the myofilaments of the sarcomere. The presence of calcium on the sarcomere causes the myosin to bind to actin in a process commonly referred to as the sliding filament mechanism, which we talked about in the last module. So we're continuing with that action potential here. Step 3 of that action potential is repolarization of the sarcolemma. So immediately after the depolarization wave passes, the sarcolemma's permeability changes again. Acetylcholine is removed from the synaptic cleft by acetylcholinesterase, and that causes the sodium ion channels to close, while the potassium channels finally open in a delayed sort of response. The influx of sodium now stops, but potassium begins to leak out so that the inside of the membrane switches back to positive and the inside of the membrane becomes more negative. This restores the charge across the membrane, also known as repolarization. However, because there's more potassium ions that eventually leave the necessary, the membrane becomes what we consider hyperpolarized. So in step 4, the sodium potassium pump restores the ion concentrations in the hyperpolarized membrane to reach the polarized conditions of the resting membrane potential. And for each ATP, 3 sodium ions are pumped out and 2 potassium ions are pumped in. Because the ion exchange is so close to equal, there is no change in the charge across the membrane as the ions are being redistributed. And during that repolarization, muscle fibers are in what we consider a refractory period when they are insensitive to further stimulation. And there are different types of refractories, and we'll talk about more of those a little bit later. You have a normal refractory period where theoretically it can't be stimulated again unless there is a large enough depolarization. But then you also have an absolute refractory period where nothing can stimulate. And as the action potentials are considered all or none responses, because once initiated, they are unstoppable. So, getting again into muscle contraction and sliding filament theory and how what we just talked about relates to that. Again, depolarization and transmission of the action potential through the T tubules. You have the opening of the sarcoplasmic reticulum and the calcium channels. You have the increase in cytosolic calcium. Calcium then interacts with troponin and causes a conformation change in that tropomyosin, which exposes the active site on that actin. The active site being exposed allows myosin heads to attach to the actin. You have ATPase activity, which helps to break down ATP into ADP and phosphate. That occurs in the myosin head, causes the movement of the actin filaments towards the Z disc. Calcium then returns to the sarcoplasmic reticulum and causes a new conformational change in the tropomyosin. This in turn blocks the active site and inhibits the actin-myosin interaction. Again, the sliding filament theory is the attachment of the myosin crossbridge within a resting muscle, which is inhibited by the presence of tropomyosin. That covers the active sites in the G-actin. Again, if calcium ions are released into the terminal cisternae by the action potential, they bind to troponin and causes the change in shape, which reveals those active sites. The troponin then pulls the tropomyosin so that the binding sites on the actin are exposed. This helps to form what we consider the crossbridge. Again, once the actin site is exposed, the high energy myosin heads attach to the actin. That's going to cause the release of the ADP from the high energy myosin head and causes the head again to pivot and bend as it pulls on the actin filament and slides it towards the midline. That also is known as a power stroke. Remember, as new ATP attaches, the now low energy myosin head will detach from the actin. ATP is then split again, and the bond energy is transferred to the myosin head and causes it to move back into the high energy position, or what we consider cocked or reactivated. So it's ready to bind to the actin binding sites once again. And when the actin potential dissipates after that, remember that the calcium is reabsorbed back into the terminal cisternae. And when that happens, the tropomyosin moves back over the active sites and covers the G-actin so that cross-bridge formation can no longer occur, which is going to cause muscle relaxation. Skeletal muscle contraction requires large amounts of ATP that may be produced aerobically or anaerobically, meaning with oxygen or without oxygen. Mitochondrial activity is the ultimate source of that energy that's required by active skeletal muscles. So providing energy for a contraction, you can have stored ADP, in which muscles store very limited amounts of ADP. It's only about 3 millimoles, which provides enough energy for approximately 2 seconds of isomeric contractions. We'll get into the different types of contractions a little bit later, but just know that's only about 2 seconds of the isomeric contraction, or about 10 twitches. So since ATP is rapidly broken down into ADP, it must be regenerated quickly. So there's a couple ways we can do that. We can use creatine phosphate, which is way more, about 20 millimoles of creatine phosphate can be stored by the skeletal muscles cells rather than ATP. However, you only make one ATP per creatine. And it's good for about 15 seconds or about 70 twitches, which, if you look back at the previous step, is considerably more. We can also get it from glycogen via the anaerobic pathways, also called glycolysis, and results in 2 ATP produced by glycogen. This process is only good for about 130 seconds or approximately 670 twitches. Although the muscles can store much more of the previous two, about 100 millimoles, the anaerobic metabolism glycogen produces lactic acid, which then accumulates in the muscle and causes soreness in the muscle, which is why you feel tired when you're exercising. You can also get energy stores from glycogen via the aerobic pathway called the citric acid cycle or the electron transport system. And this is probably what you remember from your basic biology classes, in which oxygen is required, but 36 ATP can be generated. And this process generates enough ATP for about 2,400 seconds or about 12,000 twitches. So obviously the more productive method. This is just to highlight some of the cardiac muscle, but we're going to talk more about that in the next module. You can have some disorders that are associated with this. So hypertrophy, which is an enlargement of stimulated muscles. The number of fibers within the muscle does not change significantly, but instead each muscle fiber increases in diameter. And for example, bodybuilders from exhaustive training and the use of steroid hormones. You can also have muscle atrophy, which is the lack of use of a muscle causes it to become flaccid. It loses its tone, power, and decreases in diameter. For example, a person wearing a cast for several months. You can also have a condition known as rigor mortis, which is sustained muscle contractions that occur after death due to the loss of ATP in the muscle. Without ATP, the myosin actin cross bridge cannot detach and results in prolonged contraction of the muscle, where the muscles no longer relax. And this begins about 2 to 7 hours after death, depending on environmental factors, and disappears over about 1 to 6 days when decomposition of the body starts to begin.